Good evening, my dear friends and colleagues, former President, Professor Otarnon, Vice President, Professor David Harel, our distinguished Albert Einstein Memorial Lecturer, Professor Wong. I'm happy to open the Albert Einstein Memorial Lecture, one of the two distinguished lectures we hold here at the Israel Academy of Sciences and Humanities. The other lecture is the Martin Buber Memorial Lecture, which is being held at the Humanities section. I'm delighted this evening for two reasons. The very existence of such a lecture, as we all know, Albert Einstein marks not only the utmost excellence in science, it also denotes commitment to liberal values and to humanity at large. The second reason for my delight is Professor Wong, a mark of utmost scientific excellence and of devotion to social welfare. Thank you, Professor Wong, our dear guest and Einstein speaker. We are grateful to your scientific accomplishments and social commitment, and I call upon my dear colleague, Professor Itamar Wilner, head of the Sciences section at the Academy, to present Professor Wong. Please, Itamar.
Thank you, Professor Cohen. Mr. Yongshan Chi, the representative of Taipei Economic and Cultural Office in Israel, Mr. Yuping Su, the counselor of the representative, Dr. Shimona Halprin, the former Israeli representative in Taiwan. I'm really pleased to introduce Professor Chi-Huey Wong, our Einstein lecturer. Professor Wong is the president of Academia Sinica in Taiwan, and holds a joint appointment at the Scripps Research Institute at San Diego. Professor Wong earned his PhD at MIT, and after a postdoctoral position at Harvard together with George Whitesides, he joined Texas A&M University in 1983, where he became a professor in 1987. In 1989, Professor Wong received the Hunt Chair in Chemistry at the Scripps Research Institute in the United States. And since then, he has an appointment in this institute. During the years, he acted also as head of the Frontier Research Program on glycotechnology at Riken, Japan, and as director of the Genomics Research Center at the Academia Sinica in Taiwan. The research interests of Professor Wong are in the field of chemical biology and drug discovery. He addresses a broad spectrum of challenging topics in bioorganic chemistry and synthetic chemistry, aimed to solve key medical goals related to cancer and infectious diseases. Professor Wong published over 700 papers and holds over 100 patents. He also authored several books and monographs in the area of bioorganic chemistry. Beyond the utmost scientific leadership and academic accomplishment, Professor Wong played a key role in accelerating Taiwan's biotechnology and high-tech industry. Professor Wong was recognized with numerous distinctions and awards. Besides being elected member of the US National Academy of Science and of the American Academy of Arts and Sciences, he received a long list of prestigious awards, including the Cotton Medal for Excellence in Chemical Research, the ACS Cope Award, the Nikkei Asia Prize for Science, Technology, and Innovation, and most importantly, the Wolf Prize in Chemistry in 2014. I am delighted to invite Professor Wong to present this Einstein lecture. Just a small request. Immediately after the lecture, we will have a very short ceremony where we will present Professor Wong with the formal certificate of the lecture, and I would request from all of you to attend this ceremony. Thank you.
So President Cohen and Professor Itamar Werner and Ruth, former president of Israel Academy of Science and Humanities, and my dear academicians of the Academy, and ladies and gentlemen, it's really my great honor and privilege to be this year's Albert Einstein Memorial Lecture. As you know, I have been visiting Israel for many, many times in the last 20 years. I have many friends here, and some of them are in the audience. I know quite a few scholars, and also meet quite a few government officials here. Now in Taiwan, I also often receive the visitors from Israel, particularly the scientists and the government officials, and sometimes just regular visitors. And in the past several years, I have had a wonderful experience with Shimona Heparin, representative in Taipei from Israel. And through the years, for some reason, I have a special feeling with Israel. And if I remember correctly, I will tell you two occasions which I'm still very impressed. So one is in 2008, when I visited Israel. I don't remember who was the host, but when I came to Jerusalem, I saw a lot of flags showing celebrating 60th anniversary. That was 2008. I thought the whole city was welcoming me because I was born in 1948. So I had a special feeling at that time. And the second occasion is 2 years ago, after the announcement of the World Prize in Chemistry in March. And I remember there were about 4 congressmen visit me at Academia Sinica. And I told them this is not a good time to visit Taiwan because our Congress was occupied by students. And these 4 congressmen, including the CEO of the Congress, say it's not a big deal because our Ministry of Foreign Affairs was on strike, and even our government is on strike. So, and I said, well, our Congress usually is not very effective because they always fight. And they say, no, we are even worse. We have 12 parties and we always fight, but we always have a result. And that's something I'm a little bit impressed that in Taiwan, sometimes we fight, but we don't have the result. So it's a, yeah. So as you know, Israel is a country of innovation. And a lot of people try to imitate without success. And I think on the surface, you may say, well, the country has limited resources and it faces the challenge by the neighbors. And also the country has the culture to encourage questions and accept failure. and they don't respect authority, so they always challenge authorities. But I think it's more than that, and I thought it's to do with your genetics. So somehow your genes are born with the innovation, so that's why nobody can imitate what you have today. So it's really a great honor to be here to share with you some of the research we have been doing for many years. And the title is Glycosylation in Biology. And so let me just show you what we have done in this field. And so you have some feeling about why I'm so excited about what I'm doing. So as you know, glycosylation is a reaction of adding sugar to a molecule. It's a reaction used by nature to modulate the structure and function of biomolecules. So for example, the molecule shown here, vancomycin is an antibiotic, and caractieromycin is an anti-cancer agent, and erythropoietin, or called EPO, is a glycoprotein for anemia. And then every therapeutic antibody has a shape like this. So all these molecules require the sugars on that in order to have function. And we still don't know very much about why. And this reaction is also used by nature to modify most of the proteins in our body. And this is one of the very, very complicated biological reaction, which, again, we don't know very much about that. Yeah. And if you look at that in more detail, when a protein is modified by glycosylation, for some reason, it will go to the surface of the cell and then get involved in many, many intercellular recognition events. For example, bacterial and viral infection, cancer metastasis, differentiation and development. All these processes require the sugar on the surface of cell. Again, we don't know very much about why. Yeah. A typical example is the sugars on our blood cells, which determine our blood type. For example, if you are type A, now you have the sugar on the blood cells like this. If you are type B, then you have the structure like that. Type O will be that. So everything else is the same except the sugar component. And type A blood cannot be transferred to type B because type B person has the antibody against type A. And even today, we don't know why a type A person has the antibody against type B, and vice versa. Now, the type O person is a universal donor. He can give his blood to type A or type B, but he cannot accept type A or type B blood. Again, we still don't quite understand this. Yeah. And where is the antibody from? Because type A person has the antibody against B. But where is the antibody from? Even today, we don't have a very good understanding of that. Yeah. So the process we call post-translational glycosylation is just one of the many post-translational events. But I think it's the most complex one. And even today, we don't have a good way to study this process. So in the past 20 years, my laboratory has been trying to develop new methods and tools for use to understand the biological glycosylation. And if we understand the whole thing, then maybe there will be new opportunities for application. So we have developed the methods for the making of these class of molecules, oligosaccharide and glycoproteins. And then we developed the probes, molecular probes, to monitor the process of glycosylation and to identify some unique carbohydrate structures on the surface of cells. And we developed a glycan microarray for the study of protein-sugar interaction. And these tools could be, of course, used to develop vaccines for infectious disease and for cancer, and to make homogeneous antibodies or glycoprotein with well-defined carbohydrate structure. So we know how the glycosylation or how the sugars on protein affect its structure and function and folding. And also, once we understand how the sugar is synthesized, then the enzymes associated with the synthesis may become a target for inhibition. So development of enzyme inhibitors become a very important area for research. So I will just highlight each of these to give you a flavor about what is the application of that. And the major message is that we are trying to identify the difference between the normal and the disease state. And if we understand the difference, then we may try to understand the reason why there's a difference, and then some opportunities may come. I should mention that the sugars on the surface usually are linked to some other molecules. It could be protein, so we call glycoprotein. If it's linked to lipid, we call glycolipid. So I'm going to talk about these two classes of molecules, but with the focus just on the sugar part. So the synthesis, we have developed two methods which are still commonly used today. One is the enzymatic synthesis of oligosaccharide using glycosyltransferase and coupled with the cofactor regeneration. And this method is still the method of choice if you want to scale up the synthesis of oligosaccharide. And this method actually was first conceived when I was a student at MIT. And when I was working on this area, I came across with the publication by Itama. So all of a sudden I found out my research was not that interesting because there's a pioneer there working on a related project. But I just continue to do that because sometimes you need the material to understand the function of oligosaccharide. So this becomes a very practical way of making oligosaccharide and even glycoprotein today. The second part is called programmable one-part chemical synthesis. And this was designed to make the process automatic based on the reactivity of the building blocks we designed. And so it's programmable. For example, if you want to make this oligosaccharide, and we have the building blocks here, and you start with the most reactive one, and the second most reactive, and the third. As long as you add them in sequence, then you will get the structure you want. Now, in terms of diversity, we went through the calculation. So we have about 9 monosaccharide building blocks in our body. Typically, it should be around 10. Now, if you do the calculation about how many possibilities you will get if you want to make a tetrasaccharide from the 9 building blocks, the number is 15 million. Because the linkage could be alpha and beta. And the linkage, if this the sugar to be linked to that one, it could go to this position, that position, or that position. So there are 4 possibilities. Now you calculate the possibility is about 15 million. So there's no way to go to this number there. There's no chemical or biological means today to make so many molecules. So if you don't have the access to the molecule, then you don't have the way to understand their function. So here is just one example. Suppose you want to make a molecule called global H. Now you enter the sequence to the computer, and then it will show you the building blocks. And then you just mix them in sequence according to the reactivity. And in a few minutes, you will get the product. And then after deprotection and cleaning up, you will get the oligosaccharide. So here we have 3 building blocks, starting with the most reactive one, and then the second and third. And then you get that in about 2 minutes. But here is another one. So you cannot get that. So you use that as a product. Then you enter that sequence into the computer. It shows 3 building blocks. So you mix them in sequence. Again, you get that building block. So at the end, you start with the monosaccharide. And we have about 200 monosaccharide building blocks. And that will allow you to make basically most of the oligosaccharide you want. Now the program looks like this. So you take the global H molecule and then enter that into the computer. You will do the search and it shows the 3 building blocks. And you enter that into the program and you show 3 monosaccharide building blocks. So you make this one first and then mix these 3 together and you will get the product of interest. Now, enzymatically, of course, you can do it. For example, you start with this and then use a galactose shear transferase to add the galactose to this part here. And that enzyme requires UDP galactose. And then so you come up with the regeneration of the substrate UDP galactose because it's very expensive. And you cannot use it as a stoichiometric reagent because the byproduct UDP will become an inhibitor of the process. So we simply recycle the substrate. And I don't have to tell you much of the detail because all these are published. The bottom line is that you take a free sugar and you activate that by kinase, so to make a sugar 1-phosphate, and then convert that to UDP sugar as a substrate for the enzyme. So almost every monosaccharide building block has the enzyme for that process. So if this turned out to be important, then we can make that on large scales for clinical trial or, for example, for other applications. Now, the most difficult one is sialic acid because it has a quaternary carbon center here and a carboxyl group there. So it's a dysreactive sugar. It gives you the byproduct, alpha-beta mixture and elimination product. So it's the most difficult sugar to deal with. And we cannot use it as a building block because it's a dysreactive. And as I mentioned, you have to start with the most reactive one. And unfortunately, most of the oligosaccharide have the sialic acid at the non-reducing end. So it's impossible to use that for the programmable one-part synthesis. But anyway, we come up with the building block here, and that is alpha selective. And this is based on the phosphate living group designed in our laboratory, and the Camomate here, originally developed by Takahashi and Chris. So we combine these two together and come up with this new building block for the sialidation reaction. As I mentioned, it's the least reactive. So what we did is to link that to the second sugar, so you can make a disaccharide. And now the reactivity is determined by the reducing end. And this can be considered as the protecting group of the sugar here. So now we have the building block here to cover all the sialic acid linkage. And a typical example is here for the synthesis of this important oligosaccharide. It's a cancer epitope. So you start with the disaccharide, and the reactivity now is determined by this one. So it's the most reactive one, and you mix that together, you get the product quickly. Now we can use that to understand, for example, influenza infection now. So influenza virus infect human cells through binding to the sialic acid containing oligosaccharide on the surface of human cells. And if the linkage is a 2 and 6 linkage, then it's a human virus. If the linkage is 2 and 3, linked to from sialic acid 2 position to the 3 position of galactose, then this is a receptor for avian virus. So bird flu recognize this type of sequence, and human flu recognize that sequence. But we still don't quite understand about the internal sequence. How is that recognized by human flu or the bird flu? So we use that oligosaccharide, all containing sialic acid at the reducing end, and put that on the surface of glass slide, and then use that as a microarray to see how they interact with the virus, or the real receptor is a hemagglutinin on the surface of influenza. So you can see that here is the different type of sugar, and this intensity of the fluorescence. You can see that different viruses will have a different pattern here. So you can use this to tell what kind of subtype you have been infected. And we have been using this for many, many years. For example, if you are infected by a flu virus, you can use a Q-tip to take some saliva and then put that on the surface, and then you can quickly tell what type of subtype you have infected. It's H1N1, and what type of H1N1. If it's a H5N1, you can quickly tell which one is which. And we can also use that to study the multivariant interaction of protein-sugar interaction. For example, this antibody, 2G12, was isolated from HIV patient, and that recognized this sugar, tetramannose or nanomannose, MED9 or MED4, which is on the surface of GP120. So using the array, you can measure the dissociation constant, which is about 20 nanomolar. Now, if you use the tetramannose, MED4, to compete with the bound mannose for the antibody binding, then you can determine the solution dissociation constant, and that is about 350 micromolar. So you can see a major difference between multivariant and monovalent interaction. Now, if you build a multivariant scaffold like this as an enzyme, you don't see a major difference between the two. So glycoarray is a very effective way of understanding the multivariant interaction. Now, we also look at this was in collaboration with Dennis Burton. The other antibodies isolated from HIV patient, and many of them recognize carbohydrate on GP120. So, for example, PG9 is a very unusual antibody which recognizes 2 different sugars. Usually we think antibody only recognize one antigen, but here each binding site recognizes two antigens. So it's a very unusual situation, and nobody knows what kind of structure is here, because there's no way today to produce a homogeneous GP120, or homogeneous glycoprotein, which contains only one well-defined carbohydrate structure. It's always a mixture, and because of that, you cannot get a good crystal structure of that. So this one is still unknown, but this is clear, it's a Med5. So what we did is to change the array, because the current glycan microarray on glass slide is not homogeneous. It's based on the reaction with succinimide ester on the surface of glass slide, and the distribution of succinimide ester is often not homogeneous. So we end up with the array with the sugar distribution in an inhomogeneous way, and the distance between sugar is not well defined. So what we do is we make an aluminum oxide coated glass slide, and as you know, aluminum oxide will react with phosphonic acid. So we put the phosphonic acid at the tail of the sugar, the oligosaccharide, and then attach to the surface of aluminum oxide. So we come up with the new synthetic methods to make the end-linked oligosaccharides, and this was just published in this issue of Natural Chemistry. You can see that from biantennary to tri and tetraantennary, you see this part is always the same. And then here, again, always the same sequence. And sometimes you will see biantennary structure like this or like that kind of sequence. So overall, you can just have 3 different type of modules. And you can use that to assemble about 20,000 structures of the N-glycans. And these modules were made by chemoenzymatic methods. And then after that, you can do either iodylation or glycosylation to the module and use that for the assembly of different type of oligosaccharides. And then we spot that onto the surface of the array, and you create a glycan microarray for the analysis of the antibody sugar interaction. And what we found is that the 2 sugars, one is a Med5 and the other is the 2,6 linked biantennary N-glycan. So understand this binding is important because one day we may be able to design a carbohydrate-based vaccine for HIV, because you can imagine that using the epitope to design the vaccine, you could elucidate the antibodies like this to target HIV virus. So the synthesis of the oligosaccharide led us to move into glycoprotein. Now, as you know, the so-called native chemical ligation first reported by Steve Kent and followed by Denishevsky to make all kinds of proteins and glycoproteins. But this method is limited to the use of N-terminal cysteine. And sometimes you can use a surrogate to deal with the problem. In most cases, you need to have the N-terminal cysteine. And what we did is to take advantage of the sugar, and we put the thiol group here, and to mimic the N-terminal cysteine. And as long as the sugar is within the 5 amino acid region of the N-terminal. So you can put the sugar at the N-terminal amino acid, or the second one, the third, and so on. And then you go through the same transesterification followed by ACL transfer reaction to get the product, and then you remove the thiol group. Based on the known procedure, you get the homogeneous glycoprotein. And then you can add more sugars using the enzymatic approach. And this was invented by A. Schraub Brick from Israel, and when he was a postdoc in my laboratory. He was a former student of Professor Uri Kainen, and very, very smart approach. Now we expand the horizon of for the synthesis of glycoprotein. So because many glycoprotein don't have the cysteine residue, even they have, it's limited to certain positions. So now you can basically use this strategy and combine with that to make basically almost every glycoprotein you want. But it's still limited to just 1 or 2 glycosylation sites. If you have more than 2 glycosylation sites, then it starts to get more complicated. So we use as methods to make either glycopeptide or glycoprotein, and to understand how glycosylation affects the folding and structure and function of the glycopeptide or glycoprotein. And in this case, for example, RNA polymerase II is a very important enzyme. And we know that the glycosylation will change the local structure from a random coil to a type 2 beta turn, and that induces the folding process. And a similar observation was made for EGH2 and methyltransferase. And after glycosylation at the serine 75, then it is able to fold it in the proper conformation to express the function of methyltransferase. And the methylation of lysine 27 will cause the inhibition of tumor suppression. And that is a very important target for the development of anti-tumor agent now. Now the most complex one is the N-link glycoprotein. So we want to understand what is the determining factor of this oligosaccharide on affecting the conformation of the N-link glycoprotein. So you use a human CD2 AD as a model, because there's only one glycosylation site, and there's no disulfide bond, there's no cysteine, there's no protein residue. So it's a good model to understand how glycosylation affects the folding of glycoprotein. So we made a homogeneous glycoprotein, and some to understand the contribution of each of the monosaccharide to the folding of this molecule. And at the end, what we found is that this turned out to be the most important. That is the N-acetyl glucosamine residues directly attached to the protein contribute most to the folding of the glycoprotein. So it's about 0.8 kilocalorie per mole. And then it stabilizes the folded structure by about 2 kilocalorie. And the additional stabilization comes from the second and the third sugar, and that account for about 1 kilocalorie. So these trisaccharide is essential for folding, and the rest of sugars have nothing to do with that. And they are involved in recognition. They are not involved in the energetics of protein folding. And the core fructose has also no effect on the folding. And you look at the trisaccharide, it's highly conserved from yeast to human. So they always have this trisaccharide there. And so this becomes a very important core structure for the design of homogeneous glycoprotein, because you can express the glycoprotein from yeast, and then you engineer the glycan part to make it as a human glycoprotein, for example. So then the question is about the 2 kilocalorie. Where is that from? So we look at that in details with the NMR. And what we found is that there's a phenylalanine residue nearby. And that has a hydrophobic interaction with the N-acetyl glucosamine through the 3 axial CH bond pi interaction. And that accounts for about 0.8 kilocalorie, about 1 kilocalorie. And the other 1 kilocalorie came from the remote effect. That is, when the hydrophobic interaction takes place, the orientation of the phenyl group change. And because of that change, it triggers the following hydrophobic interaction, and that takes care of the additional 1 kilocalorie. So overall, it has a 2 kilocalorie energy involved in the folding of this important glycoprotein. And then we use this result to design an influenza vaccine. And so we take as you know, if you take a flu shot, the antibody generated usually are targeting the hemagglutinin part of the influenza. So a lot of people try to use hemagglutinin as the epitope to design influenza vaccine. And Ruth has a many very interesting and breakthrough result in this field. So what we did is we look at the glycosylation again. And what we found is that position 27, well, I should mention that their hemagglutinin is a trimer, and each monomer has a 6 glycosylation site, and all of them are end-link. So 27 is essential for folding, and 142 is for the binding specificity. If you don't have the glycosylation at this position, the binding specificity will change. And the other 4 sites are not important for folding. And the other observation is that the sequence, the protein sequence around the glycosylation site, that is the sequence covered by the sugars, are highly conserved. So for some reason, the virus is so smart, they try to escape from the host, the immune system, by glycosylation of their sensitive part using the host glycosylation machinery. Sugar were added by human, and the virus take advantage of that glycosylation from human to protect themselves from the attack by the human immune system. So what we try to do is to remove the sugar, as long as they don't affect the conformation of hemagglutinin. And we did that, and we make the monoglycosylated hemagglutinin using endoglycosylates to remove the outer part of the sugar. And that turned out to be very effective vaccine. And the immune response generated antibodies is able to neutralize many subtypes of hemagglutinin. So it points to a new direction for universal influenza vaccine design, and that is as long as you expose the highly conserved region and the immune system is able to detect that, and it will generate antibody against influenza virus. You may ask why the antibody will recognize the native structure, because sugar usually is pretty flexible. So once the antibody is there, it's able to slowly push the sugar away and then bind to the protein. So we have a lot of experiment to show that it actually is the case. So here's another example. You take an antibody, therapeutic antibodies, for example, Rituxan is a very effective antibody for lymphoma. And if you look at the glycosylation of the antibody, it contains about 50 different types of glycoforms. And some are very good, some are very bad. But in average, you see the activity there. So what we did is just to remove that to make all the sugar to make the monoglycosylated one. And then we add the sugar based on the method I just showed you, using the N-glycans we synthesize. And we test the activity of the glycoform here. We found that this sequence with the 2,6 linkage here is the best, and you will increase the activity by about 100-fold compared to the commercial therapeutic antibody we know today. So antibody-dependent cellular cytotoxicity is enhanced by the use of the right oligosaccharide, because the effector function is determined by the glycopeptide in this region, and that the interaction of the Fc region with the Fc receptor is affected by the sugar at the position 297. So antibody has two functions. One is recognition of the target, and then after that is the effector function, which is used to kill the target, and that requires the proper, the accurate structure of the glycan there. So if you take for example, the universal antibody against the human influenza developed by Novartis, it's called FI-16. You simply change that sugar to the glycan I just mentioned, the 2,6 linked biantennary glycan. Now you can see that the killing effect will increase by about 2 or 3 folds. And in the challenge study, you can see that it's also much better than the reported one. And Ebola also, we know that the antibody against Ebola is very effective from tobacco. You remember that? And it's not effective from CHO cells. And I think that is to do with the glycosylation. So we take the antibody from CHO cells, and again, we re-engineer the glycan to make that into the 2,6 linked biantennary glycan, and again, it's very, very effective. So the last part of the lecture will be the identification of a cancer-specific glycan epitope. And as I mentioned, you need to find a unique structure on the surface in order to be used as a cancer vaccine. So the epitope selection is very important. I think it has to be non-cell and specific. That's why most of the vaccines are against infectious disease, not cancer. Now, overexpression of epitope is not sufficient to be used as an epitope for vaccine design, because self-antigen will cause autoimmunity. And the immune checkpoint interference will take place, like a PD-1 will be induced to shut down the immune system. And carbohydrate-based vaccine will encounter additional barrier. That is, carbohydrates usually are T-cell independent. So they don't have memory. You go to B cell directly and to generate IgM. As you know, IgM has no long-term memory. So it's important to deal with that problem if you want to use carbohydrate as a vaccine. You have to have a class switch, or you have to link carbohydrate to a carrier protein so it can go through the T-cell pathway. So here is the strategy. Often people link carbohydrate to a carrier protein and use it as a vaccine. So it will go to the MSC pathway. If you go to, it's taken up by dendritic cell, for example, and then it's processed by protease, and then glycopeptide will be presented to killer T cell. If it's presented by MSC2, then helper T cell will be the target, and then after the release of the chemokine or lymphokine, you will tell the B cell to make the antibody. Yeah. But even that, you still generate IgM as the major class of antibody. So we need to have a class switch in B cell from IgM to IgG through recombination. And that can be done by, for example, interleukin-4, because B cell has an interleukin-4 receptor. And once it recognizes IL-4, you will go through the class switch from IgM to IgG. And that is done through the design of a new adjuvant. We use a dendritic cell surface CD1 receptor and design the glycolipid adjuvant. And once it interacts with killer T cell, you will go to Th1 and Th2 pathway. And alpha and gamma represent Th1 pathway, that's the adjuvant pathway. And then IL-4 represent Th2, it's an immunosuppression pathway, but it's also inducing the class switch. So it's a balance between Th1 and Th2. We have, and that is to do with the affinity of the, or ability of the glycolipid with CD1D and T cell receptor. So we now is able to design the adjuvant to have the right balance of Th1 and Th2 for the adjuvant activity, and at the same time to induce the class switch from IgM to IgG. So to look for the unique carbohydrate epitope, then we feed the cells with this or that. And these are for fucose, and that's for sialic acid. We know in cancer cells, they are highly glycosylated or sialylated. So we focus on these two sugars. And once the cell takes that as a substrate, they will go to glycolipid or glycoprotein. And then we add another probe, and that will react with the alkyne to form the triazole. And once the triazole is formed, fluorescence is induced. So it is based on Sharpless chemistry, but it's a click-induced fluorescence approach to minimize the background signal there. So using this one, we are able to identify the unusual sugars on the surface. Every time when we do this, we found that this pathway shows up in cancer cells. And we don't see that in normal cells. So I call this a global series pathway, starting from SHCL3, and after sialylation, give you SHCL4. After glycosylation, will give you global H. So these three glycolipids are cancer specific. And of course, they are found on embryonic stem cell, but they disappear when the embryonic stem cell differentiate. When we have cancer, they come up again. So we don't understand why, but this is the observation. And the enzyme, the key enzyme is this one, beta-galactosyl transferase 5. We call beta-3 GALT5. And this, again, is highly expressed in cancer cell. So once this enzyme is there, then the global series glycans shows up in cancer. So these are the three glycolipids there, and they are only found on cancer cells. And we look at 15 different types of cancers, and they all have these three. And we don't see them on normal cells. And you can see that from breast cancer, triple negative cancers, cervical cancer, brain tumor, kidney, liver, ovarian, pancreatic, prostate, lung, mouth, and colon. All the major cancers have these three unique glycolipid markers on the surface. And then we ask the question, are these three on the surface of cancer stem cells? And the answer is yes. So if you use SHCA3 as a marker, we can enrich cancer stem cell. If we use that together with the two non-protein markers, then you can see that only 10 cells will be enough to enrich, to isolate the stem cell. And if we use the current markers there, then 1 out of the 264 cell will be the so-called cancer stem cell. So SHCA3 actually is a very important marker for breast cancer stem cell. And then we knock down the enzyme beta-3-galactify. So the 3-glycolipid cannot be expressed on the surface. Then the cancer cell go to apoptosis. They commit suicide if the 3-glycolipid are not there. And we now understand that once the enzyme is knocked down, caspase-3 becomes active, and that will destroy the kinase FAK. And once that is destroyed, the signaling cannot go through. So the cell will commit suicide. And this is linked to the 3-glycolipid, but the detailed pathway is still not very clear. And we use the antibody against either one of the three, and they show very effective again. And if we modify the sugars, as I mentioned before, using this, I would call it universal glycan for antibody, it will maximize the activity. So because it will interact with the Fc gamma 3 receptor, and that will activate the effector cell to kill the target cells. So all these seem to point toward the direction that these three global serious glycan are cancer specific, and they may be able to be used as an epitope for the vaccine design. So we again conjugate global H to the carrier, and then we use that designed adjuvant to induce a class switch. It's called C34 glycolipid, and we immunize the animal, and then the antibody generated was profiled using the glycan array to see if they really recognize the 3-glycolipid target. So to our surprise, the vaccine is able to induce the antibody to recognize the 3 global serious glycan. So one vaccine will target 3 cancer epitopes, and very clean. And this one is the adjuvant I talk about that is used to not only serve as an adjuvant, but also to induce the class switch from IgM to IgG. And we look at that in more detail and found that when the vaccine is taken up by dendritic cells, it's processed. The fucose is cleaved. So that's why you see the antibody is also able to recognize not only global H, but also SHCA3 and SHCA4, because all these have a common epitope, that is SHCA3. Now, in B cell, it's not processed, so the IgM is more specific for global H, and they don't recognize 3 or 4. Now, if we want to make the sugar even more immunogenic, then you can introduce the Azide group to this position or that position. Because in our experience, in our lifetime, we never experience Azide. You take any food or drug, none of them has an Azide group there. It has fluorine or nitro or whatever, but we never get into the azide. So it turned out to be a very good immunogen. If you modify the sugar, one of the sugar functional group with the azide, then it will become much more immunogenic and will trigger the immune system to respond to that. And the antibody, of course, cannot find the azide on the sugar, so you will just attack the related structure that is either global H or SHGL4, I just mentioned. So how do you see the difference between antibody and vaccine? As you know, antibody and vaccine belong to the so-called target therapy. And if you want to use antibody as a drug, it's called passive immunotherapy. So the patient has to have the target. Otherwise, the antibody is not going to be effective. So for example, Herceptin has to be used for the patient who are HER2 positive. And only 20% of the patients are HER2 positive. Yeah. PD-1 is another antibody, very popular now, and about 20% of the patients are responsive to a PD-1 antibody. And vaccine has another hurdle, that is, you need to have the immune response to generate antibody. Otherwise, it will not be effective. So if you don't have the immune response, there will be no efficacy at all. So if you use a vaccine, you ask the question whether there's an immune response, and then whether the patient has the target. And then from there, you can compare the treatment group with the control and to see if the vaccine is effective. So what I can tell you is that the glycosylation glycans are cancer specific. And they are only found on cancer cells, not on normal cells. So they are good target for vaccine design. Of course, they are also a good target for antibody and also for the small molecule inhibitors like beta-3 or GALT5. And more than 15 different types of cancer can be used for the treatment. So the first generation of global H vaccine was developed by Denischowski at the Memorial Sloan. And in phase 1, it was shown to be mainly IgM, but it has a CDC activity there. So it's not a long-term, has no long-term memory, but it's effective. And the second generation is the one I just mentioned. It's designed for long-term memory with the IgG response. But this one has gone from phase 1 to phase 2 and phase 3. And the result, phase 3 in Taiwan was just disclosed. And it shows very effective if the patient has an immune response to the vaccine. And I think this probably is much more long-lasting and could be very effective for early stage or even for preventive purpose. Yeah. So these are all what I want to share with you, and just want to thank the people involved in the work in the old days. These are the people at Scripps. Particularly, I want to thank Timo and Ezra for their contribution. Timo was involved in the programmable one-part synthesis. And most of the work I talk about today was done at the Academia Sinica. And particularly, I want to thank Eric Ma and Zhongyi Wu and Alice Yu for the collaboration on the glycan array and the vaccine design. So it takes so much time to share with you about most recent result in our laboratory. And thank you very much for your attention.
So,
this is the Einstein medal.
contain scroll and
It's in Hebrew.
it's in Hebrew and English. First of all, we have to take a photo. And I want to tell you about the scroll. It has inscription, of course, and decoration. And the inscription says as follows. The Israel Academy of Sciences and Humanities hereby expresses its appreciation to Professor Chi-Huey Wong for his presentation of the Albert Einstein Memorial Lecture for 2016. This occasion serves to commemorate Albert Einstein, the man and his accomplishments. Albert Einstein's interests and influence were not restricted to theoretical physics, but embraced a spectrum of areas of profound significance for science and society. In the spirit of Albert Einstein, Chi-Huey Wong has enriched science and society in many ways. Now, the decoration depicts a candelabra, which was presented at the temple, which has 7 candles, and it marks divine knowledge and light, and pomegranates, which marks plenty, and the menorah, the candelabra, and the pomegranates are, of course, the signs, the marks of the Israel Academy of Science and Humanities. So, thank you very much.
Thank you.
It was a real pleasure.
Thank you.
Thank you, Itamar.
Thank you.
Thank you, Itamar.
And thank you all. Thank you for coming.